What really happens when we eat proteins? Do they work directly in our body, or are they rebuilt into something new? This fundamental question touches the core of biochemistry and nutrition. Let's explore what actually happens. First, let's understand what happens in your digestive system when you eat a cooked steak or boiled egg. When you cook protein at temperatures above 60 degrees Celsius, the heat causes denaturation. The protein unfolds and loses its three-dimensional structure. This process is irreversible. The native folded protein transforms into a denatured, unfolded state. Here's what happens. A tightly folded protein structure unfolds into a linear chain when heated. This is why egg whites turn from clear to white. Here's the critical question. Once these proteins are denatured by cooking, can they refold in your body and perform their original function? The answer is definitively no, and understanding why reveals fascinating biochemistry. Let's examine what your digestive system does to these proteins. Your stomach produces the enzyme pepsin in a highly acidic environment with pH around 2. At pH 2, this is 10,000 times more acidic than neutral water. Pepsin cuts the protein chain at specific points, breaking it into smaller pieces called peptides. Then, in your small intestine, more enzymes break these into individual amino acids. In your small intestine, enzymes like trypsin and chymotrypsin complete the job, breaking everything down to individual amino acids. The pathway is protein to peptides to individual amino acids. This process is nearly 100% efficient. Studies show that 95 to 97% of dietary protein is completely broken down into amino acids before absorption. Why does your body go through all this trouble to break down proteins? Why not just absorb them whole? Your intestinal barrier has tight junctions between cells that only allow molecules smaller than about 600 Daltons to pass through. A typical amino acid has a mass of 110 to 180 Daltons, which can pass through. But even a small protein weighs 10,000 to 100,000 Daltons, far too large to cross the intestinal barrier. Small amino acids can pass through the tight junctions, but large proteins are physically blocked. This is a crucial protective mechanism. If intact foreign proteins entered your bloodstream, your immune system would recognize them as invaders and mount an attack. This would cause severe allergic reactions and potentially life-threatening anaphylaxis every time you ate. So what happens to all those amino acids after absorption? This brings us to the amino acid pool concept. Your body maintains a pool of free amino acids in the blood and cells. Amino acids constantly enter from digestion and leave to build new proteins. At any given time, your body contains about 100 to 150 grams of free amino acids in this pool. Every day, your body breaks down and rebuilds about 250 to 300 grams of its own proteins. This is called protein turnover. Here's an interesting question. If we turn over 300 grams of protein daily, but typically only eat 80 grams, where do the other 220 grams come from? Answer: your body is incredibly efficient at recycling. Most of those 220 grams come from your own proteins that are constantly being broken down and rebuilt. Now, let's see how your body uses these amino acids to build brand new proteins. The ribosome is like a molecular factory. It reads mRNA instructions from your DNA and assembles amino acids in the exact sequence specified by your genes. This is the central dogma of molecular biology. DNA is transcribed to mRNA, which is translated to protein. This is crucial. Each protein's amino acid sequence is determined entirely by your genes, not by the food protein you ate. Let's take a concrete example. Building hemoglobin, the oxygen-carrying protein in red blood cells. One hemoglobin molecule contains exactly 574 amino acids in a very specific sequence. Your ribosome can synthesize one complete hemoglobin molecule in about 90 seconds, adding roughly 6 amino acids per second. Let's address a common misconception about food enzymes working in your body. Some claim that eating raw foods preserves enzymes that can help with digestion. Let's examine this scientifically. Reality: any enzyme from food is immediately denatured by your stomach acid at pH 2. Most food enzymes work optimally at pH 6 to 8, close to neutral. But your stomach operates at pH 2, which is 10,000 to 1,000,000 times more acidic. A food enzyme entering stomach acid is like dropping a delicate origami structure into a shredder. It's instantly destroyed. And even if an enzyme somehow survives stomach acid, it would still be digested into individual amino acids in the small intestine before absorption. Are there any exceptions? Can any intact proteins cross the intestinal barrier? Yes, newborn infants have a special temporary ability to absorb some intact antibodies from breast milk during the first few days of life. This provides passive immunity, but only works for about 24 to 48 hours after birth. Then the intestinal barrier matures and closes this pathway. The intestinal permeability to proteins drops by about 90% within 48 hours after birth. In healthy adults, less than 0.1% of dietary proteins might cross intact, and these usually trigger immune responses. Let's look at the energy and efficiency of protein metabolism. Breaking down proteins through digestion requires energy from ATP molecules. It costs approximately 4 to 5 ATP molecules to break each peptide bond during digestion. But building new proteins costs even more energy. It takes about 4 ATP molecules to add each amino acid to a growing protein chain. You might wonder, why does the body break everything down and rebuild it if this costs so much energy? Answer: the specificity and safety benefits far outweigh the energy cost. Your body needs proteins built exactly to your genetic specifications. What does all this mean for nutrition and health in practical terms? First, cooked versus raw protein has essentially the same nutritional value, because both are broken down to amino acids. Second, expensive enzyme supplements you take orally are simply broken down like any other protein. They don't work as enzymes in your body. Exception: lactase supplements work in your intestine to digest lactose, but they're not absorbed. They just work locally in your gut. What actually matters for nutrition is the amino acid composition of foods, not whether the original protein was functional. All protein sources, meat, eggs, beans, nuts, end up in the same amino acid pool. Your body can't tell if an amino acid came from a cow or a chickpea. Not all amino acids are equal. Let's discuss essential amino acids. Out of 20 standard amino acids, 9 cannot be synthesized by human cells and must come from diet. These 9 are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Your body can synthesize the other 11 amino acids from other molecules, like glucose and nitrogen sources. This is why protein quality matters. Foods must provide adequate amounts of all essential amino acids. 9 amino acids must come from food, while your cells can manufacture the other 11 internally. Let's summarize everything with a complete answer to our original question. Food proteins absolutely do not restore their original functions in your body. This is impossible for multiple reasons. They are completely broken down to individual amino acids during digestion with 95 to 97% efficiency. These amino acids join your body's amino acid pool, where their origin is completely lost. Your ribosomes then build entirely new proteins using your own genetic instructions, not the structure of the food protein. This system ensures all your proteins match your species requirements and individual genetic specifications. The journey is complete. Food proteins become amino acid building blocks, which your body uses to construct your own unique proteins according to your DNA blueprint. This system represents elegant biochemical engineering. It provides safety, specificity, and efficiency all at once.